Hey guys, welcome to the last and final week of biology. I know this isn't how we all expected the school year to end, but I've had a great time being here with you guys, and thank you so much for tuning in every week and watching my presentation and what I have to tell you or help teach you guys. So this is the last and final week, like I said. We're talking about natural selection, which we finished up last week, and then we're going to try to cram in a little bit of classification with you guys too, because that's one of the funnest things that I think I do in my classroom, and I wanted to share that with you guys. So we talked about some of these words last week, and the ones I really want to pay attention to are one, Heidi Weinberg, and we're going to talk about stabilizing, directional, disruptive selection, and what these founder and bottleneck effects are. Now, last week we talked about how evolution is change over time, okay? So how do organisms that are in our world adapt to their environment, and how do they, in these adaptations, are they able to survive and keep basically living and keep going on, okay? So there's some ways that this happens in the everyday world. And Heidi Weinberg states that allelic frequencies remain constant in a population that's genetically equilibrium. Yeah. So that sounds like science, doesn't it? Okay, let's break this down because you know what this means. Alleles. We talked about alleles. We've talked about alleles for 3 weeks. Alleles means what version of the gene do you have? So brown hair, blonde hair, red hair. What color eyes? Tall, short, fast, slow. Whatever these genetic traits of these genes that we have in our DNA, there are different versions of them, and that's what we mean. And frequency means how often. So how often are these versions of the genes stay constant? In other words, is a trait increasing or is it decreasing or is it staying the same? And staying constant means it's staying the same in genetic equilibrium. So basically the Hardy-Weinberg principle says that there's no evolution going on. Population stable. There is no changing, no adaptations going on. All right, and again, this was review from last week. We talked about genetic drift. Genetic drift is that you've gotten some type of drift. You had a small population, some new trait got introduced, and we saw a shift in the amount of traits that we had. That's what we mean by genetic drift. Founder effect, though, is basically when the original population becomes isolated. So, way back when, maybe today, we see volcanic eruptions happen every so often. Maybe something happened, it burned the ground, all these fires in Australia, something happened. Organisms got trapped somewhere and they became isolated from the rest of the group. Something happened that isolated them, okay? And maybe it just so happened, let's just say we had insects again, red and green insects, but maybe green's the common color. The majority of the insects are green. But in this isolation, for whatever reason, the population ended up being mostly the red ones. You're going to see the shift in the population where all of a sudden now the most common color you see is going to be red. And that's what we mean in founder effect. We had an isolated population happen, shift in these genetics, shift in the traits, and you kind of see something that's not so common become common again. Bottleneck, also, we're talking about, you hear about extinction of animals, or the number of animals are declining. So something has happened that has caused the numbers of these animals to decline over time. And because of that, again, you have kind of shifted which traits are there, all right? So this wide variety of traits, maybe when we talk about, I mentioned moss last week, you know, in a city environment, there's lots of soot because of pollution and stuff. So these white moss don't hide very well. And, you know, they all died off. We were bottlenecking, and it was choosing for these dark moss that were able to, you know, hide and camouflage themselves. Gene flow. Again, this is a closed system, but basically gene flow happens when you have some random event. And by random event, this is where we talk about a tsunami or hurricane. We know all about hurricanes down here in Alabama, okay? That volcanic eruption again. Something happened and it came through and it really just kind of wiped out that environment. All right, mutations. I don't know how much we got into mutations right before the break at the end of the third quarter, but mutations happen every day as organisms replicate in our DNA, okay? And whether or not that mutation survives, but that is a way to introduce evolution. Now, here's where I really want to get on the crux. We have stabilizing, directional, and disruptive, okay? So we're going to talk about each of these. when we're going to talk about stabilizing, we have usually there's two traits. Let's just go, I'm going to use the moss for an example. Okay, now this didn't exactly what happened, but this is what would happen in a stabilizing selection. We have white moss, and at the opposite end we had the dark black moss. If we were in a stabilizing selection over time, you would see the population shift to something in the middle. So maybe like a gray moth, something in between. In real life, a good example of stabilizing population is birth weight. Okay, let's look at infants. When infants are born with a really, really low birth weight, they don't survive too well. They do because of the medical capabilities we have now because of doctors and hospitals and stuff. But if in the everyday world, if you weren't able to get medical treatment, low birth weight babies typically don't survive so well. Same thing, opposite end, really large over birth weight babies sometimes also have some complications to deal with depending upon what's going on. Typically, you see the stabilizing direction going on that really there's a nice healthy range for birth weight. And that's what we see in the everyday environment. So stabilizing means you don't see anything at this end or the opposite end, but you see something in the middle. Directional, you see a shift, okay? So let's go back to our moss. We had white moss and we had black moss. And what do we talk about? The white moss were really easily seen in the city environment. And so birds were able to eat them. The black moss survived. You saw a shift in the population to this end down here all the way to the right. So in the, in a directional selection, you see a shift towards favoring one trait or the other. Okay, you see the shift. Then we have disruptive. Something happens and you actually see both traits survive. And there's a very distinct line. There's nothing in the middle. There's no middle birth weight. There's no gray. You would see white moss and dark moss on this particular one. Okay, so again, stabilizing shows you a middle. Okay, directional shifts from one direction to the other, and disruptive means you end up with a complete two different populations of organisms with these two different traits. All right, so we're about to go into classification, but before we do, it's time to win a prize. Okay, so my question for you guys is, from last week we talked about Darwin, and Darwin went on this great big trip around the world, and he went to the Galapagos Islands, and he was able to make all these observations in order to come up with this theory of evolution that organisms change over time. So my question is, what was the name of the ship that Darwin was on? All right, if you know the answer, send an email to mcpsstv@mcpss.com, and let's see, let me know you got it. All right, guys? Good luck. All right, so for organisms, I want y'all to think. There are millions of organisms across, billions, I said millions, even more, billions known of organisms. Okay, believe it or not, we've only discovered about 5% of them. All right, that's still a whole lot to figure out and how to name and classify. Now, we're still finding stuff, especially like in the Amazon, are really one of the biggest places. If you're interested in science and you want to be somebody to discover a new organism, learn how to scuba dive, learn all about oceanography. That's one of the biggest unknowns we still have. So think about that for your future. But we had to figure out how to classify things, how to group organisms, and usually we group them based on similarity. Now, this name for classification, and there are people that actually, that is their job. They name and classify organisms, and that is the study of taxonomy, and somebody that does that is called a taxonomist. Okay? Now, why do we do this? Well, we got to figure out how to do it accurately and uniformly, because sometimes there's some misnomers. Now, down here, so a misnomer, that means it's named something that it's not. Have you ever seen a seahorse? Is a seahorse a horse? What about jellyfish? If you hadn't taken zoology yet, guess what? A jellyfish isn't a fish. It's actually something what we call a cnidarian. So this is what we mean by misnomers. Our common names aren't actually always accurate. Okay? And then there's also confusion in our names. Now this is a cougar, but depending upon where you're at in the United States, it's also called a puma. It's also called a mountain lion. It has about 10 different names depending upon what region you're in, and they're all the same animal. So we really had to figure out, could you imagine trying to figure out if you were like, well, I discovered a puma, and somebody's like, but I discovered a mountain lion, and guess what? Y'all discovered the same animal. So how do we figure out how to keep these things in order? So when we know when we see something, it's not something new or unique, it's actually something that's already known. And that's where we get to Linnaeus. Linnaeus is the father of taxonomy. You know, he was so good at his job that nobody's figured out a better way to do it. And, you know, in science, that's kind of rare because science is always evolving. I mean, even like what we know about cells and DNA and proteins now, it's still changing and we're still learning. We're still using Linnaeus's way, okay? And his way is called binomial nomenclature. Binomial, bi means two, nomial means name, so it's a two-name system, two-name naming system. All right, and we use the genus and the species. And when you do this, you can use Latin or Greek. If you are printing on a computer, you always italicize it when you're going to put the scientific name down, just FYI. If you are handwriting it, guys, if you ever have to write a paper for your teacher, you should underline the scientific name to let them know that that's the true scientific name. And then the genus is always capitalized and the species is always lowercase. So the example, believe it or not, here's bird. I love this name, Turdus migratorius. But you can see here, T capital, that's your genus. M lowercase, that's your species. That's the American robin. Check it out. All these guys are bears. And if you look, guess who is more closely related? All right, we've got the panda, polar, and grizzly bear. Look at the genus. If they have the same genus like the polar bear and the grizzly bear, Ursus, they are more closely related, okay, in similarities. And then the species part tells you exactly which Ursus bear we are talking about. Now, there is a hierarchy to naming. When we start grouping things, we're going to group them first by domain. And there are 3 big domains, okay? There is archaea, there is bacteria, and there is eukarya. Okay, now eukaryotic, that should sound familiar. It's cells that have a nucleus. So everything in the eukaryotic domain has a nucleus inside their cells. Then we have bacteria, which are bacteria, and then we have archaea, which is just ancient bacteria. Then we start breaking down into kingdoms. Well, archaea has the archaea kingdom. The bacteria domain has the bacteria kingdom. Our domain that we're in, because we're eukaryotic organisms, has 4 kingdoms. These should sound familiar. Plant, protist, fungus, or animal. Okay, so then we go kingdoms. Then we start going into phylums. All right, now and then we go into class, order, family, genus, there's the first part of our scientific name, and species, the second part. So this always goes for the broadest to the most specific species. Okay? Now, if you have trouble remembering this, because I guarantee you there's usually always a test question somewhere, guys. At some point in your life, you're going to get the test question. Put these in order, from broadest to specific or specific to most broad. It doesn't matter. But did King Philip come over for good spaghetti? And if you can remember that little acronym, you will always be able to remember the order of your hierarchy for taxonomy. All right, so again, domain is the broadest, species is the most specific, and then here's a good example. If you look at this top, we have up here, we have the grizzly bear, black bear, panda, red fox, squirrel, coral snake, and sea star. Those are all animals. But we're going to go into the chordates. Okay, we lost the sea star because they don't have a backbone. They have their own phylum. Take zoology your junior or senior year and you'll learn all about it. Okay, or marine biology. They cover the starfish too. After chordates, we go down to mammals. We lost the snake. Snakes are reptiles. They're not mammals. Okay, then we go down to carnivore. Carnivore means that they eat meat. All right, if we have lots of squirrels down here, squirrels don't eat meat, squirrels eat nuts. Okay, then we go into Ursidae, and if you look, now we have the 3 bears. We've lost the fox, so we're getting even more specific. All right, then again, we talked on that other slide how the grizzly bear and the polar bear are closer related. This is genus Ursus, and then down here, we're sticking with the polar bear at the very bottom, Ursus arctos, and we have our polar bear. Okay, so we went from the broadest from the kingdom all the way down to the most specific. And here's another one, but did King Philip come over for good spaghetti? Now, dichotomous keys. This is how you classify stuff. All right, we always start with questions, okay? And your questions are always in pairs, and they're always yes or no answers. And it can be something as, does it have 6 legs or did it have 8 legs, like insects versus arachnids, okay? So yes or no. So here's an example. So we're going to start down here with this guy, okay? This one on the bottom. And question 1, tentacles are present. Tentacles are absent. Yes or no? Are these tentacles? Yeah, they are. Okay, so it says go to 2, so you go to 2. Are there 8 tentacles? That is a whole lot more than 8 tentacles down here. So it's not the octopus. Is there more than 8? Yes, so you go to 3. Now, do these tentacles hang straight down? Do those hang just below it? No, they don't. So tentacles upright, that is the sea anemone. So our dichotomous key, di means 2, so it's always 2 questions, key, takes you through a series of questions of yes or no, and it will help you identify the organism that you're talking about. Now, in this case, as you can see, every time you get to the no, it has an answer. Now, if I was going to go back over here to the octopus, you don't start down here where we left off at question 3. You go right back to the very first question. So, let's do this again. Tentacles are present. Yes. Did it have 8 tentacles? Yes. Question 2. So, we're on the octopus. So let's have some fun with a dichotomous key. Everybody loves Disney. These are some of my favorite Disney characters. Some of them you may know, some of them you may not know, but that's okay. Now, if you're going to do this, like I said, when you're going to make your own dichotomous key, because this is something I always give to my kids to do in my classroom, you start with a question that tries to divide in half. Now, in this particular case, I started with, was it a boy or a girl? Was it a boy? If it's a boy, yes, and I grouped them, right? That was my first question. Was it a boy? Yes or no? And then I grouped them accordingly, okay? So we're going to stick here with this because we've only got 3 organisms, or 3 characters to talk about. The next question. Now, you can pick any question you want. Just remember, it has to be a yes or no question. I chose, did it have a bow? And we have yes, and we have no. Now look, underneath my no, we have 1 organism. When you get down to that 1 organism, that's when you name it. Okay, so in this case, we're down to 1. That's Clarabelle. Now I still have 2 over here. What is a question I can ask, to differentiate between these 2? Well, polka dots are kind of my thing. So I actually asked, does it have polka dots? And the answer is, if it was yes, it's Minnie, and if it was no, it's Daisy. I've now isolated those between their own individual. All right, so if you were to go back, I have over here, first, how to do it, okay? So this is kind of, I know it's a little cluttered. I wasn't sure how much space I could go over, but these were the questions I asked. Is it a boy, yes or no? And I wrote, okay, 6 of them were yes, 3 of them were no. Did it have a bow, mimicking what we just saw in the PowerPoint? 2 of them were yes, 1 was no, and when I got to 1, I named it. Okay, so there's my Clarabelle. Then I got to polka dots, yes or no? Yes, it was Minnie, no, it was Daisy. All right, well, that took care of that side. Well, now I'm going to go over here, and let's start. I had 6 boy characters left to name. Tail, yes or no? Well, that divided them into 3. 3 of these guys had a tail. 3 of them did not. Sorry, I forgot to change it here. It's not tail. It was hat. Did they have a hat, yes or no? Out of the 3 that had a hat, did it have a bone? 1 had a bone. We have now identified Pluto, and then 2 did not. Out of those 2, notice, I don't go back and go to everybody else. You stick with the group you're working in. So I'm just talking about now these 2 that had a hat but don't have a bone, and I asked who has big ears. And one of them was Mickey, because Mickey has big ears, and the other one was Dale. And if you're not familiar with Chip and Dale, you'll have to go watch some cartoons. They're cute. Now we can go over here. I had my 3, all right, and I asked who had long ears. Well, this is Goofy, and then I had 2 that didn't have long ears that actually had the hat. All right. asked who had a bill. Well, that would be Donald Duck, okay? And so once you've gone through and you've kind of laid out your chart for who has, and you've identified everybody based on your questions, you can make your key. And in this case, this was question 1. This was question 2, and I kind of follow my arrows. This was question 3, and that ended that. And then we came down here, 4, 5, has a bone, 6, whoops, sorry, that's not 6. This is 6, keep going down the aisle, okay, 7, and 8. And you can number them however you want, but then you go write it, okay? So, question 1, is it a boy? Is it a girl? Question 2, has a bow? Does not have a bow. Question 3, had polka dots? Does not have polka dots. And after you've written down all of your questions, because you went through and counted them, you've got your questions written out, yes and no versions, then you can map it out. So, let's go. Is it a boy? If I said yes, well, this pointed to question 4. And I said, go, so you write, go to 4. All right. Is it a girl? If you had said, no, it is not a boy, it's a girl, this pointed to question 2. And you write, go to 2. And you go through this all the way. So let's go for has a bow. So bow, yes. That went to question 3. But remember, does not have a bow. That ended over here. We didn't have another question. We got down to 1 character. And so that's where you don't have to write go to. You just write whatever your organism is. So I know that's a lot. Have fun with some dichotomous keys. You can find a lot of them on the internet, guys. There are some, if you just Google dichotomous keys, and there's some interactives you can do. I always have fun. Go outside, pick a bunch of leaves from plants, all different types of plants, and make your own. Start classifying. Green, red. A good time to do this is in the fall, too. But I've had a lot being here for the last 6 weeks. I hope you've enjoyed it as much as I have, and I look forward to seeing everybody in the fall. Thank you.